Our next speaker is Dr. Michael Wunsch. Michael is a resident plant pathologist here at the Carrington Center. He's originally from Montana. He received his PhD at Cornell University, and he's been here since 2010. His emphasis is disease management in broadleaf crops, in particular both pulse crops and oilseeds. So, and Michael has a 70% research appointment and also 30% extension appointment. So Michael has 4 stops. He has the most of the tour tonight by one person. So this first stop, he's going to talk briefly about a product for management of sclerotinia in soybean.
This field behind us was purposefully managed to try to establish sclerotinia. This field behind us is a large experiment to test the efficacy and also to evaluate the optimum application timing, rate, and method for Contans. Now, Contans is a commercial formulation of the biological control agent Coniotherium militans. Studying this product is particularly difficult in field research setting, because in this case what we're trying to do is we are trying to study the control of the formation of the little mushrooms on the ground, okay, right below the canopy. So if you all know, the sclerotinia fungus produces these black resting structures known as sclerotia. Now, the resting structures can persist in the soil for many years, and they germinate to form little mushrooms called apothecia, and the apothecia are your only spore-generating source. And this is actually the reason why biological control in this sense makes sense, because what you're doing here is you're applying the biological to the soil to degrade the resting structures. If you have fewer resting structures, you're going to have fewer mushrooms. Fewer of those little mushrooms, you have fewer spores, and you should have less disease. This disease is different from many of our other diseases in that it does not produce spores on diseased tissue. So secondary spread of the disease is limited only to plant contact and not to secondary production of spores. So if we can really limit our initial production of spores, we should get quite a bit less disease. What we did in this case is that we set this up as a normal small plot trial. And we had, for instance, our non-treated check adjacent to, say, a high rate of Contans. Okay, in that non-treated check, you should get lots and lots of the little mushrooms, producing lots and lots of spores. Okay, immediately adjacent, you'd have the Contans treatment, and if the Contans is working, you should have a lot fewer mushrooms, a lot fewer spores. Okay, and in theory, you should get less disease. But if those plots are side by side, the spores move. Okay, and so your spores from your non-treated check will wipe out your plot next door. Okay, and so in order to get around this problem, what we did here is we set up our plots 60 feet apart throughout this field. You see it on a grid. And our first year doing this, and what we do is we actually spread sclerotia in those plots. Okay, and our first year doing this last year, we were able to show very pronounced differences in the survival of those sclerotia and the production of the little mushrooms, depending on the application rate, timing, and method of the Contans. Okay, but we're not able to show any treatment differences in terms of disease, because in this case, I figured that there wasn't that much sclerotinia in the field. Was wrong. Many times the sclerotinia was worse outside our treatment plots, and basically we got a lot of sclerotinia moving in from outside. Okay, and so we were able to show degradation of the sclerotia by the Contans, but unable to quantify disease reduction and yield response. And we really need to quantify that. Okay, so this year we applied Contans between our plots. Okay, to try to standardize the environment between our plots. We ran into a different problem though. We applied sclerotia to our plots, and in this case, those sclerotia had been in our warehouse for a couple of years, and they were no longer viable. And so as a consequence, what we have out there are not enough sclerotia to be able to differentiate treatments. Very frustrating. To get around this problem, next year, we're moving to huge plots. Okay, the trial next year will be on this field over there. We're going to move to plots that are about an acre in size. Okay, so that entire acre will be treated exactly the same. With that size, it won't matter whether added sclerotia are viable or not, because with that size, just our native sclerotia alone will be enough to differentiate treatments. We should not have to worry about neighboring treatments diluting our effects, because if we take our disease assessments and our yield from the middle of those plots, we should be sufficiently insulated from the neighboring treatment. And so what we're looking at is, it depends, we're going to have to decide which spray rig we go with. We're looking at plots that could be as large as 240 feet by 240 feet, okay? And obviously we're looking at fewer treatments then. This will be geared for conventional tillage. So next year, stay tuned, we should be able to have really large plots where we can rigorously assess the disease control and yield response associated with using Contans under conventional tillage. I think you can see quite convincingly, looking at this field, you see all those plants with the wilt symptoms, that even in a dry year like this one, if we irrigate aggressively, we can generate results. And so next year, hopefully, we'll have some data for you, at least the first year, in a database to hopefully inform your decision making whether this is a product that would work for you.